directions that our lab is taking in terms of clinical proteomics. We want to really use these proteomic technologies to try to move it into a clinical setting. And I think one of the easiest, most applicable settings, which is a very low bar in terms of clinical applicability, is actually microbiology. And we are systems biology lab. We not only do proteomics, we are doing genomics and transcriptomics. And here is a nice illustration of how we are actually combining next generation sequencing and mass spectrometry in order to solve some of these diagnostic dilemmas in microbiology. So I'll talk about how, as of today, mass spectrometry is used for microbial identification in a discovery setting, and how we are using next-gen sequencing and also targeted mass spectrometry to not only diagnose bacterial infections, but also figure out antibiotic resistance. And in this particular case, I'm going to talk about carbapenemase resistance. And I'll show you how we are using similar approaches of whole genome sequencing and proteomics, including targeted mass spectrometry to diagnose mycobacterial infections. So microbial diagnosis using mass spectrometry is already in clinical use. So almost 14 years ago, when I was setting a lab, my own lab at Johns Hopkins, I made one key decision. I said we are not going to have multi-top mass spectrometry in our lab. And my reason was that we don't want to do peptide fingerprinting, which is what it does. There's another form of mass spectrometry which uses MALDI-TOF that does give sequence data. But here we have, 2 years ago, you have MALDI-TOF that is approved, first by European FDA and then by US FDA for microbial identification. And I was saying to myself, almost 14 years ago, we made the decision not to use this technology because of the several problems. And yet, in the world of microbial diagnostics, it is useful enough. And I will tell you that now that it is used, what are still the problems with this? It relies on measurements of intact protein masses. Most of the talk that we have talked about, when people say proteomics today, they almost imagine that you know that is sequencing of peptides. It is a bottom of proteomics. Here, we are not talking about peptide sequencing, we are talking about protein masses. And we all know how heterogeneous proteins are, and proteins carry many different post-translational modifications. So we are looking at that fingerprint, and that is often matched with a library, and the library is often not generated by the investigator. The library is generated by the company that sold the mass spectrometer. That means if the people at the company have not done research into it, you cannot benefit from that. The other issue that has is you're relying on a company to do the research for you, and you essentially are believing their data. The reason it is very useful in the clinic and has gotten wide acceptance is, according to me, there are two reasons. One is it's cheaper. It is not using the kind of fancy technology that I'll talk about. So for $200,000 or so, you can get these machines, whereas the mass spectrometers we use cost $1 million today. Not to say that those costs won't come down. But the other is, it's not so much in the actual running of the samples, but it's in the way the results are presented to the clinician. Most clinicians, I think it's fair to say, do not understand mass spectrometry. So they have to be basically told what is positive identification. So there's a lot of mumbo-jumbo that goes into converting the answers that go from this matching to a yes or no answer, whether or not you have identified or not identified an organism, and the companies have successfully done that. This is actually not possible today in most of the other types of proteomics, where we argue about whether a phosphorylation site was real, whether a peptide was identified. Here, we are actually saying, in a clinical setting, this is identified. So that ease of use is there for the clinician. It's considered definitive. That's one take on it. But really what bothers me is, that there are lots of issues with both false positives and false negatives. And often we are relying on data given by the manufacturers of the mass spectrometers about what those numbers are. And I think this is becoming, this is increasingly being recognized that you cannot tell antimicrobial resistance, and I will tell you how a different kind of mass spectrometry can actually do it today. And this is not universally applicable to all microbes, whereas when you have sequencing-based approaches, you can essentially address any microbe. And I feel the future is high-resolution mass spectrometry for microbial identification, which is based on sequencing of peptides. And in one experiment, you can actually sequence thousands of peptides. And then there are other easier platforms that can be used, which are highly multiplexable. And these are targeted mass spectrometry platforms of multiple reaction monitoring or parallel reaction monitoring. And although we are talking about proteomics, I think we cannot forget that there are many other ways to come to this conclusion. And here I will tell you about some of those technologies. And the major one there is whole genome sequencing. So at the protein level, we are looking at the product made by genes. But what if you could just sequence the genes by themselves? Well, today, for about $100 or so, you can sequence a whole genome of a microbe. And so I think in the future, we are going to be using whole genome sequencing and sequencing-based mass spectrometry in the clinic for microbes. And I have to admit that for many, many years, I avoided this area of research in our lab because the whole time, it was just too easy. The bar was so low. Because we are trying to conquer cancer proteomes, this is nothing for a proteomics lab. But given that it has still not caught on in a clinical setting, we decided that we are going to push the envelope to show proof of principle experiment that this can be done in a clinical framework. And now we are working with manufacturers of these mass spectrometers to say, how can we take your instruments into a clinical setting? Because I think they need this help. And I think we also discussed some things about FDA, and that's why I was trying to clarify that FDA does not mandate. What we are hoping is that by working with these companies, we will actually develop data that can then be submitted to FDA for them to actually make a call whether or not this can be used. These instruments can be used in that kind of a pipeline. So here we have a culture of the microbe. So this is when from a clinical sample, you grow the microbe in a microbiology lab. And here we have a pressure-based lysis. This is a barocycler where we extract the proteins using a very high cycling to 45,000 psi back to room pressures, which is about 10 to 15 psis. And then we do a very rapid trypsin digest. So this is still very conventional workflow for proteomics. We are doing bottom-up proteomics here. We use these high-resolution mass spectrometers, and we are doing sequencing. So we have basically done a proof of principle study where we have taken a set of clinical strains and reference strains of Klebsiella pneumoniae, and some of them were sensitive while others were resistant to carbapenems. And that's because they make carbapenemase, which target carbapenems, which are broad-spectrum antibiotics. So these have beta-lactam rings, and they're relatively resistant to hydrolysis by most beta-lactamases. And I think the reason why this is clinically a very important problem is carbapenemases are used clinically in patients who are hospitalized as pretty much a last agent in these patients who are very, very sick. And there are many, so although this is one word, actually there are many different enzymes within this class of carbapenemases. You've got class A ones, and I'll talk about these. These are Klebsiella pneumoniae carbapenemases, KPC 1 through 8. Then you have metallo-beta-lactamases, and I will talk about VIM and NDM. For NDM, for example, many of them are named after the place where they were isolated. So this is New Delhi metalloprotease. And also others which are class D serine carbapenemases like oxacillin, hydrolyzing, or oxa-beta-lactamases. So from our collaborators at Johns Hopkins, we got this list of samples where we had one which was carbapenem susceptible, we had a clinical isolate, and we had others which are essentially given by CDC, Centers for Disease Control, which are known to be KPC 3 positive, KPC 2 positive, VIM positive, NDM1, and OXA positive. And our goal was to use next-gen sequencing and mass spectrometry to try to tell them apart. So what we have done is we have taken these cultures, we have digested them into peptides, and then we have sequenced them. And you can say, so this is, again, often when we are looking at cancer proteomes or tissue proteomes, we are running 50 to 60 different runs from one sample. Here, we are running the entire proteome as a single run, and I'll tell you, we are also running that for a short time. So it's the biggest shortcut we could ever take in proteomics. And in that shortcut, we have identified 800 or so proteins. So these genomes have about 4,000 or so proteins. We have sequenced 500 proteins in this short time and about 2,500 to 3,000 peptides. And for example, KPC3, this is now a plasmid encoded protein. And we have sequenced 8 different peptides that uniquely come from this. This is the kind of data that you don't get in current settings. We are getting, so each one of these peptides that come from this resistance marker, we are getting from this MS spectrum. So we know we have this peptide because we have sequenced it. This is OXA181 encoded peptide, and again, this peptide maps back to this carbapenemase. And in order to see how we can do this in a clinical time frame, this is the one thing that is highlighted by manufacturers of these MALDI-TOF platforms is, well, you need to get answers really fast. And many of these LCMS type regimes, they take hours. So what we did was, well, we will digest the material for 1 minute, and we'll run it for 1 minute. We'll digest it for 5 minutes, and we'll run it for 5 minutes. And in all of those, we can uniquely identify the organisms very, very easily. So within a 5-minute time period of digest, so that means this is a 10-minute processing. 10-minute processing, and we pretty much have instantaneous results. We are talking about a very, very small genome. In 10 minutes, we can basically figure out what is happening. And here, we can identify that it is Klebsiella pneumoniae for sure, but also that it makes this protein that is the resistance marker. This is a susceptible isolate where under the same parameters we identify the organism, but there is no peptide that is identified that comes from KPC3 plasmid. So this is using a proteomic approach. One could imagine that the definitive answer lies at the level of genome. So we have also done whole genome sequencing where we have multiplexed all of these samples into one lane of MiSeq next-gen sequencer. So it's a small sequencer, and these genomes are 1,000 times smaller than human genomes. So it's a trivial problem even at the next-gen sequencing level. So we are sequencing 10 genomes at the same time and getting anywhere from 50 to 90x coverage of every base pair in the genome. And I will come back to this. And here we basically say, okay, all of these were identified as Klebsiella pneumoniae. And I will come back to this, where one of them, which are supposed to have this resistance marker in Klebsiella pneumoniae, we didn't really find that it mapped. Something was up. One of them was called as 1 encoding New Delhi metalloprotease 1 or NDM1. When we did whole genome sequencing and mapped back to the reference strains, it turned out that it didn't have NDM1, it has OXA181. But this is a reference strain that has been provided by CDC to our colleagues at Hopkins. We went back with this data to them, and we said, it doesn't make sense. You called it NDM, and we see it's OXA181. They go back and look at their notes, and they say, we are sorry, very sorry, but it was actually, you are right. We don't need to be told we are right. We have the answer. We are telling them that you were wrong. Okay, so that's one. Remember this one here. We had this that did not map to Klebsiella at all. This was even more confusing. So we went back to them and said, what is happening? Because when we then search against all the bacterial species, we see that this is one category of E. coli. So here, and we have 90% coverage, and this is classified along with some of the other known disease-causing E. coli. They then went back to their notes, and they said, oops, we are sorry, this looked like Klebsiella pneumoniae. Now we agree that it was actually E. coli. We had to do genomics to tell them this, but also our proteomics for this particular one, we also saw that it did not map, and none of the peptides map. So by genomic methods or by proteomic methods, we would have told them, this is Klebsiella pneumoniae they are talking about, not, this is E. coli they are talking about, and not Klebsiella pneumoniae. So here we were using discovery proteomics, where our goal is to sequence as many peptides as we can in a short time and be able to tell what is the organism we have and what resistance marker they may have. Here what we did was, we have already identified the organism, so we know peptides, and we have peptides we have sequenced from the protein that encodes for antibiotic resistance. So what we have done is we have developed these targeted assays now, where the question we are asking is, are these peptides present in the sample? This is an even easier experiment, and we can multiplex them. So what we did was, we chose some peptides which come from outer membrane protein, and this essentially says that this is Klebsiella. Then we chose peptides coming from each one of those resistance markers, KPC3, KPC2, OXA181, WIM1, and WIM2, and we are looking for these peptides and their fragments in a targeted fashion. And here, this is the outer membrane protein that is common to all of the Klebsiella isolates except E. coli. Of course, we don't see it in E. coli, the one that was mislabeled by them. They thought even this was Klebsiella. So in this targeted assay, we could have also told it is not Klebsiella. Now, we have these examples where we found this KPC3 and one that they had mislabeled. These are OXA181. These are negatives. So in a targeted fashion now, we can pick up these resistance markers where we know the question we are asking. So one of the caveats to this is we are still dealing with clinical material from a microbiology lab where we have lost 1 day. They have already taken the specimen, they have grown it in the lab for 1 day. And that is the time they are going to take to do their series of biochemical tests. If they want to do any further testing, they will take 1 more day. So after 1 day, if we do whole genome sequencing now, that protocol takes 24 hours. It's one of those rare things where I would still say whole genome sequencing is actually the definitive assay. But that takes 24 hours. The proteomics assays that I talked about, they take 10 minutes per sample. So that means in 1 day you have the material, the microbiologist is starting to think what it is, and in 10 minutes we subject it to this kind of mass spec. We have the answer of what it is and what resistance it encodes for this very important class of last resort agents, carbapenems. So 1 day and 10 minutes, we have the answer. If we did next-gen sequencing, we'll have it in 2 days. Well, in that setting, 1 extra day may be too much. But now when we talk about tuberculosis, here, time is not that big a problem because this Mycobacterium tuberculosis grows very slowly. It takes 4 to 6 weeks to culture it and to do drug sensitivity and all of that. Here, we can now start to take advantage of the fact that 1 day in sequencing is nothing in this 4 to 6 week time frame. And I feel the future here is going to be some combination, again, of whole genome sequencing and proteomics. If you have sputum, you might be able to directly do mass spectrometry. It may be easier in certain settings. But otherwise, what whole genome sequencing might offer you is not just for identification, but also to try to tell you what drugs these patients should be put on. Almost 5% of all incomers at most hospitals in a country that is almost endemic with TB, India, have drug-resistant tuberculosis. There's one referral hospital in Mumbai in India where they tell me that 60% of cases they have multiple drug resistance TB. So you can imagine it's a very big number and it's rising. What I feel is, and precious time is wasted because people cannot figure out what are the drugs they should be put on, because there are many different kinds of resistance patterns that are already known, and if you knew them, then you might be able to put them on second line and even third line therapies. And the genome sequence actually gives you that choice. Here I'll just start with some proteomic data where what is often done is you take sputum samples and you do acid-fast staining that tells you that maybe this is Mycobacterium tuberculosis. And often this is done by a technician who may or may not be so well versed in these methods, and many of them turn out to be negative. And many clinicians in these endemic countries, they actually don't care about the negative results. What they say is, clinically, I still think my patient has TB, and they put them on presumptive therapy. Wouldn't it be nice if instead of having these false negatives, we actually could convert these negatives into positives? And here what we did was, we took some of these sputum samples with presumed TB, and we can identify all of these proteins that are unique to Mycobacterium tuberculosis. There are many atypical mycobacteria, but these peptides come from pathogenic Mycobacterium tuberculosis. So proteomics can take you directly from sputum into a diagnosis of MTB. You don't need a microscope. You are getting the sequence of these peptides that tells you. So I would say we need to get into molecular diagnostics. This is molecular. Microscope is not molecular. But then, what about this? Often when you identify, people are put on presumptive therapy, which is standard therapy. But then, what about drug susceptibility? It's not done at many, many centers. And often what ends up happening is the patient has to fail therapy, which means after 6 to 9 months of taking these drugs, they come back and the symptoms are still there. Then people might resort to doing this if it is not part of standard pipeline. Another way is, can we do something upfront? This is a study that our colleagues at Johns Hopkins just published, and what was interesting was that while they were testing a method which gave them a quick peek into some sequence variants that are associated with resistance, they had some genotype-phenotype discordance. That means when they do a drug susceptibility test, they can tell that a given isolate is either resistant or sensitive to a drug. But when they do this microarray-based testing, it is either false resistance or it is false susceptible. And what we said was, let's look at them and see what genomics can tell us. So we have taken one isolate that is sensitive to all anti-TB drugs, and 9 with this discordance, and one non-tuberculous mycobacterium, Mycobacterium abscessus. And we did whole genome sequencing on the same platform. And again, we can take advantage of the fact that it's fast, it's cheap. We can do 10 different genomes in one run. And here, I will now show you what I meant by this genotype-phenotype discordance. So they had done these drug susceptibility testing, and they knew that this isolate was surely resistant to INH, all of these 3. They had this array method by which they were looking for mutations in these 2 genes where known mutations are known to correlate with the resistance, and they could not find any. So this is where we have false resistant, because the actual test tells you it's resistant, but molecular testing by their method tells you it's sensitive. So the inference for molecular testing is sensitive, but phenotype testing is resistant. So interpretation is false sensitive, and when we did, these are results from our whole genome sequencing. And here, what we can see is clearly that CATG gene has a deletion. And that deletion we can easily pick up in this strain. This one here, 42, we have a deletion there, and that deletion actually truncates this gene into a very small protein product, and that is what is causing it to become resistant to INH. When you go to this other one, again, it's resistant by drug sensitivity testing, nothing identified by array, but then we identified a point mutation in CATG gene, and this is easy to say that's 41. So that's the mutation. So again, molecular testing by whole genome sequencing is telling us this is the third one. Again, we have this mutation that is captured very, very easily by whole genome sequencing. And that's why I'm saying that in the future, if we really care about these patients, these technologies have brought down the cost of testing to low levels, and they are only going to drop. They are not going to increase. And here we have a case where it was sensitive by drug testing, but when they used their array, they actually found a mutation. And this did not make sense because this mutation is correlated with resistance. When we do the sequencing, there is actually no mutation. So when you use a low-resolution method like an array, you find a mutation, but that is an incorrect call. When we do sequencing at 100x almost for every base pair, now we can tell it is wild-type sequence. So again, this was sensitive by phenotype and actually sensitive by whole genome sequencing at that position. And what we would like to be able to do is also a quick screen to definitively identify MTB, because cultures take 4 to 6 weeks. And here again, we designed these assays where we could target these peptides that come from Mycobacterium tuberculosis. And this is now using parallel reaction monitoring. And in these 4 cases, we can easily see that this peptide that comes from MTB is there. And these 2 samples come from this other atypical mycobacterium, Mycobacterium abscessus, and we don't see it. So again, you have a nice differential diagnosis built in there. And then when we use a peptide that is made by Mycobacterium abscessus, but not MTB, we see in these 2 samples, but not in the others. So using a targeted fashion, you can maybe build these plus-minus assays much more readily rather than mass spec sequencing based. So that's a shortcut to clinical use again. And one can set up very, very standardized regimes to develop quick diagnostics based on this. So this is just for diagnosis. In Klebsiella, I told you we can also monitor resistance markers. Here we are doing more research into how we can catch the peptides that correspond to mutant protein products. But we can use whole genome sequencing to, again, jump to the conclusion of whether or not the strain that has infected a particular human being or a patient is supposed to be resistant. So here what we have done is, this is work that we have done in India, where more than 200 samples with all different kinds of resistance phenotypes have been collected, and we have done whole genome sequencing for half of them. And we can easily find mutations in these isolates, but I think what is more telling is that here what I plotted is these are the drugs, and for each drug I've named the different genes whose mutations can lead to resistance. And these gray bars are the number of isolates, and these yellowish orange color is showing how many new mutations that are not in well-established databases have been identified in this patient population. So we have, of course, identified many of the known mutations that confer resistance, but we have also identified new mutations. So this is one of the issues where anytime you use an array-like platform or a panel can only look at a few mutations. But we are still very early in the process of looking at these genomes. So we have many more mutations that can cause disease, and we need to increase the repertoire, and the only definitive solution is not a panel today, but whole genome sequencing. And when you work out the economics of looking at, let's say, 50 to 100 mutations, it sounds so bizarre, but looking for 50 to 100 mutations in this context is more expensive than doing the whole genome at 100x coverage. It's so counterintuitive, but we have to realize this as we move. This is 2016, and I think we should behave as if we are in 2016 and not use primitive ideologies and technologies. We can do whole genome sequencing. Surely in a country like the United States, there is no excuse not to be doing this. It is not in clinical use today. So we have to do it as a research project, show the clinical utility that obviously we argue about so much about what personalized genomics is doing in the context of a much more difficult disease called cancer. There are so many mutations in every cancer genome. There are 50 to 500 mutations where sometimes it's not even known what is the driver. We are talking about much simpler problems. Infections are still the only curable diseases we have. We can cure them, but we've got to put them on the right drugs. And I think this is our shortcut. So, and this is a summary of all the data, and it's mind-boggling. So we found 90 or so known mutations, 110. So the number of new mutations we identified are actually more than all the known ones. But what is even more shocking to us and our collaborators, and we have gone back, and I told you about some problems we had with our microbiology lab at Johns Hopkins. Johns Hopkins is the #1 hospital in America. We have problems in 10 cases, 2 of them had issues. Now we are talking about third world countries. Here, in 23 cases, we were given samples that were labeled as Mycobacterium tuberculosis. Our sequencing tells is non-tuberculous mycobacteria. What is also known is that co-infections do occur, but I think people don't have a good handle on that. Well, using these next-gen sequencing technologies and proteomics, we can tell them easily apart from each other. So here, 23 is too high for our collaborators. So we really don't know whether it is the technique, it is some handling, some growing in the microbiology lab, where exactly these problems are happening, or it is truly the case, in the case that the patients have NTM that are misdiagnosed. So we are not saying they're misdiagnosed. All we say is that the material that we have is non-tuberculous mycobacterium. Okay. Now, we have every sort of problem that you can imagine that we have already encountered in a small set of 100 cases. So just bear with me here. So, according to what was known based on the drug testing, these are supposedly drug-sensitive isolates. They were given to us saying this is sensitive. We sequence the genome and we find drug resistance associated mutations. We are talking about mutations that are documented to be associated with drug resistance. So they are being falsely called drug sensitive. Okay. Now, there are patients who are being treated with ofloxacin, and we basically find in those MTB isolates from those patients that they have a mutation that tells us they are not going to respond. And this is not the first-line therapy. So they're already put on the second-line therapy, but they actually already have mutations to that. Who's going to take care of that? And my hypothetical question to people is, if you had an infection with TB, would you spend $100 to actually do whole genome sequencing? Again, in the US, it's a rhetorical question. It's a no-brainer. But the issue is, how do we deploy it in third world countries like India? And also, 1 out of every 5 cases was also infected by an NTM, and these non-tuberculous mycobacteria are very hard to treat, and that is just well known. And so finally, I think going into the future, this is my last slide, what I've shown you still is the easy space for all of this to operate in, for either genomics or proteomics, because we have isolates. We have these material obtained from cultures. We are now pushing the technology to do it directly from specimens. This was done from cultures. We want to go to specimens, and the specimens could be blood culture, CSF, sputum, pus, bronchoalveolar lavage fluid, urine. And then for proteomics methods, there's one very special case, for example, C. difficile, where if you have the bug, even if you have the genome encoding a toxin, you have the RNA, people would like to actually see the toxin, which is not easy to assay for. So again, proteomics methods can come to the rescue to see when is the toxin actually being made. And I would like to thank all of these people who worked with me on this, and thank you for your attention. Correct.
Yeah. And I want to know what is
Yes, so what I was talking about was what are the limitations of a MALDI-TOF mass spectrometry method, and the last point on that slide was that MALDI-TOF based methods, so MALDI-TOF mass spectrometers are used in clinical microbiology today, but it is not for every organism, mainly because these companies that make the mass spectrometers, but also LabScan themselves, take the organism, generate the profile, and put it in a database. If it is not in the database, then you cannot identify it, because it's based on a fingerprint, not sequencing of peptides. And an example is we had a collaborator, and I was talking to Dr. Ravi Kumar also. So he came to us because he wanted to identify a fungal pathogen. And that fungal pathogen was not in the database. So we said, we will do discovery proteomics where we don't rely on the library, but we basically can sequence multiple peptides and can tell you which fungus it is.
And the other question was that in one of the Klebsiella strain, you saw that the genome analysis or nucleotide sequence was exactly the same as E. coli.
Yes.
That surprises me.
It was labeled, so it was a clinical specimen, and based on the standard protocol in the microbiology lab, they had presumptively identified it as Klebsiella. And they gave it to us because we were really excited about using these methodologies. So when we ran our assays, we see that by genome sequencing, it is not Klebsiella, and by proteomic sequencing, it's not that. We went back and we told them we are puzzled because we didn't know the answer. All we told them was it matches to this particular strain of E. coli. Then they went back to their notes and they said, aha, we think this is where we made. They had a morphological, it was more slimy, and based on the phenotypic characteristics, they identified it as Klebsiella. And they can say that, and they said it does fit this particular subtype of E. coli.
Were those sensitive to that antibiotic?
Right, okay, so that's a very good point. Because if you have the diagnosis wrong, there's a good chance you're using the wrong drug to treat them. So this is the other issue, and now I've been talking to many other people, and I think these types of errors are quite commonplace, but once clinical labs get established, you have an identification. The clinician treats, if the patient fails to get better, only then more things start to kick in, and people may do more culture and more sensitivity and all of that. Maybe it will be discovered, maybe it won't be. But surely the patient may be put on an incorrect antibiotic in some cases, but not in all. Many times they're given broad spectrum drugs where they will kill Klebsiella and they'll kill E. coli. So it depends.